think about the precision of modern medicine or the resilience of our food supply biotechnology is increasingly the engine driving these advancements.
Right.
And if you've ever wanted to understand the fundamental how behind it all, well, you've definitely come to the right place today.
Exactly. We've pulled together quite a bit of material that really dissects the core principles and the processes of biotechnology.
Okay.
Our aim today is to basically pull out the most crucial, the most interesting bits and give you a clear, organized understanding of modern biotech.
So we're diving deep. We've got excerpts explaining the bedrock science, things like genetic engineering, bioprocess engineering,
Right.
the fundamentals.
That's right. Chapters 11 and 12 from our sources are going to be our main guide here.
Okay, let's get into it then. This field, it's huge now, but where did it actually start? What's the origin story?
It's interesting to look back historically. The sources mention a big shift in thinking, maybe around the 17th century. Figures like René Descartes. There was this growing idea of using science, the natural sciences, to develop tech that improves human life, comfort, welfare.
A very human-centered view.
Absolutely. And that really shaped how we looked at the natural world, how we studied it.
Okay, so using knowledge for human benefit. And biology, even way back then, that was mainly about food, wasn't it?
Primarily, yes. The biological world was our main food source, always has been. But then biotechnology comes along in the 20th century.
And that changed things.
It was a massive leap, a qualitative jump, really. It didn't just increase quantity, it improved the quality of health and food production significantly.
Okay, so fast forward to the 20th century. Biotechnology, it's a term we hear all the time. But what does it actually mean? What's the core definition?
Well, in the broadest sense, it's just using living organisms, or maybe enzymes from them, to make useful products and processes.
Simple enough, like old traditions.
Exactly. Think about making curds from milk, or baking bread, brewing wine. Those all use microbes. That's biotech in a way.
So my kitchen experiments count.
Your grandma's kitchen. Basically the original low-tech bioreactor. It's fascinating. We just scaled up those same basic biological ideas.
Right, but modern biotech is more focused, isn't it? It's not just letting yeast do its thing.
Yes, exactly. While those traditional methods use natural processes, the modern definition, the one used more strictly, often involves genetically modified organisms, GMOs,
Ah, okay.
and usually on a much larger scale. It's about deliberately changing the genetics for a specific purpose.
Got it. So manipulating life at the genetic level. Can you give some concrete examples of this modern stuff?
Sure. Things like IVF, in vitro fertilization, test tube babies,
Right.
or synthesizing genes from scratch and using them, developing DNA vaccines, even trying to correct genetic defects. These are all modern biotechnology.
Wow, okay. It's broad.
It is. The European Federation of Biotechnology, the EFB, they have pretty good definition, they call it, the integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services.
Integration. Okay, that captures it well.
Mhm.
So what were the key breakthroughs, the core techniques that made all this modern stuff possible?
There were really two core techniques that enabled modern biotechnology. First one, genetic engineering.
Okay.
This covers techniques to actually alter the chemistry of the genetic material.
Hmm.
So DNA and RNA,
Changing the code itself.
precisely. And then introducing that altered material into host organisms. The goal being to change their phenotype, their observable traits.
So direct editing of the blueprint, that's powerful. What's the second technique?
The second is bioprocess engineering. This is more about the how on a large scale, maintaining sterile conditions.
Sterile. Why is that so critical?
Well, think about making something like antibiotics or vaccines. You need to grow huge quantities of specific microbes or cells, right?
Right, you don't want contamination.
Exactly. Bioprocess engineering ensures you're only growing the desired organism in sterile conditions, in large vats, basically. It's chemical engineering principles applied to biology, maintaining purity.
Okay, large-scale, super clean factories for cells. Got it. Let's circle back to genetic engineering then. How did we even get the idea we can manipulate genes like that?
That's a great question. The sources point to looking at natural reproduction. Sexual reproduction, for instance, naturally creates variation.
Shuffles the genetic deck.
Right. You get new combinations of genes, some good, some maybe not. Asexual reproduction, on the other hand, mostly just copies what's there. Less variation.
So nature mixes things up, but traditional breeding, like for crops or animals, that can be a bit messy, right?
It can be, yes. Traditional hybridization often brings undesirable genes along with the desirable ones. You want a bigger fruit, Maybe you also get weaker disease resistance. It's a package deal sometimes.
A bit of a lottery.
Kind of. Genetic engineering, though, things like recombinant DNA, gene cloning, gene transfer, that lets you be much more precise.
How so?
You can isolate only the gene you want, the desirable one, and introduce just that specific piece of DNA. You avoid dragging along the unwanted baggage.
Much cleaner. Okay, but say you isolate a gene, a piece of DNA. If you just stick that into a cell, what happens? Does it just work?
Usually, no. A random piece of DNA floating around in a cell won't typically multiply on its own.
It needs to be part of the system.
Exactly. To be copied and passed on to daughter cells, it generally needs to become part of the host's own genetic material, like integrating into a chromosome.
And why is that integration needed for copying? What's the trigger?
The key is something called the origin of replication, or ORI. It's a specific DNA sequence on a chromosome.
The starting line for copying.
Precisely. That's where the cell's replication machinery kicks in. So if you want your foreign DNA to be copied, you need to link it to an origin of replication.
Ah, okay. Hook it up to the cell's copy machine.
Right, and when that happens, when the cell copies its DNA, it also copies your foreign piece. That's essentially what cloning means in this context, making lots of identical copies of that template DNA.
Okay, so link your gene to an ORI, put it in a cell, and the cell does the copying. This sounds like the basis for that first big breakthrough, right? The first recombinant DNA.
Absolutely. The creation of the very first artificial recombinant DNA molecule was a landmark. The idea was to take a gene for antibiotic resistance
Okay.
and link it to a naturally occurring plasmid from the bacterium Salmonella typhimurium.
Plasmids, so those are the little extra circles of DNA in bacteria.
Exactly. Small circular DNA molecules that can replicate on their own inside the bacteria, separate from the main chromosome. They have their own ORI.
Got it. And who are the key players here?
This was Stanley Cohen and Herbert Boyer back in 1972. Real pioneers.
What did they each bring to the table?
Well, Boyer was working with restriction enzymes in E. coli. These are the molecular scissors we mentioned. He discovered they cut DNA at very specific spots.
Right.
And crucially, they often leave these little single-stranded overhangs. He called them sticky ends.
Sticky ends, love that name. Why sticky?
Because they naturally want to pair up to hydrogen bond with a complementary sticky end.
Ah, okay. So Boyer had the scissors. What about Cohen?
Cohen was the expert on plasmids. He'd figured out how to isolate these plasmids from bacteria and, importantly, how to put them back into other bacteria.
Okay, so one had the scissors, the other had the vehicle and the know-how to move it around.
Exactly. The breakthrough was combining these. They used Boyer's restriction enzyme to cut the antibiotic resistance gene out of one plasmid and also to cut Cohen's plasmid open at a specific spot.
Creating compatible sticky ends on both pieces.
Precisely. Then they used another enzyme, DNA ligase, you can think of it as molecular glue,
Okay.
to join the antibiotic resistance gene into Cohen's cut plasmid.
Wow. Cut and paste, but with DNA in a test tube.
That's essentially it. Creating a new combination of DNA, a recombinant DNA molecule, in vitro, outside a living cell.
And then what?
Then they introduced this new engineered plasmid back into E. coli bacteria.
And because the plasmid had its own origin of replication,
It started replicating inside the E. coli, using the bacteria's own machinery. And every time the plasmid replicated, it copied the antibiotic resistance gene along with it.
so they were cloning the gene.
Yes, they called it cloning of antibiotic resistance gene in E. coli. It was the proof of principle for the whole field.
Incredible. So from that foundational experiment, we can kind of map out the basic steps for modifying any organism, right?
Yeah. Our sources lay out 3 core steps that follow logically. First, identify the DNA you want, the bit with the desirable gene.
Find your target.
Second, introduce that identified DNA into your host organism.
Get it inside.
And third, make sure that introduced DNA is maintained in the host and, crucially, passed on to its offspring, its progeny.
Maintain it and make sure it's inherited. Okay, sounds simple laid out like that, but the how must be complex. Let's dig into the tools needed for this. The tools of recombinant DNA technology. We already met restriction enzymes.
The molecular scissors.
Yeah.
Absolutely critical. They were first found back in '63.
As a defense mechanism.
Yeah, in E. coli. Bacteria were using them to fight off viruses, bacteriophages. One enzyme added methyl groups to the bacteria's own DNA to protect it,
Okay.
and another one, the restriction endonuclease, chopped up the invading virus DNA.
Smart defense. And Hein 2 was an important early one.
Right, Hein 2. Isolated about 5 years later. What was key about Hind-2 was that its function depended entirely on recognizing a specific DNA sequence.
A specific pattern of bases.
Exactly, a sequence of 6 base pairs. It always cut the DNA at that specific point within that sequence. That predictability was huge.
So each enzyme has its own specific target sequence. How many do we know of now?
Oh, loads. Over 900 restriction enzymes identified for more than 230 bacterial strains, and each one recognizes its own unique sequence.
Wow. Is there a system for naming them? E. coli, Hind-2?
There is. It's quite logical, actually. The first letter is the genus of the bacterium, the next two are the species, then maybe a letter for the strain, that R for RY13 and E. coli, and finally a Roman numeral for the order was discovered in that strain.
Genus, species, strain, order. Got it. E. coli from Escherichia coli, RY13. Makes sense. Now, these belong to a bigger class called nucleases.
Yes, nucleases are just enzymes that cut nucleic acids.
Yeah.
There are two main types, exonucleases and endonucleases.
What's the difference?
Exonucleases nibble away nucleotides from the ends of a DNA strand. Endonucleases, like our restriction enzymes, make cuts within the DNA strand.
Endo for within. Okay, so restriction enzymes are sequence-specific endonucleases. How do they find their target and make the cut?
They basically scan along the DNA molecule, like reading it. When an enzyme finds its specific recognition sequence, it binds there.
Locks on.
Right. And then it cuts the sugar phosphate backbone of both DNA strands at specific points within or near that sequence.
Double strand break. And you mentioned these recognition sequences are often palindromic. What does that mean for DNA?
Think of a word palindrome, like Malayalam, reads the same forwards and backwards. A DNA palindrome is similar, but it applies to the two strands. The sequence of bases reads the same on both strands if you read them in the same direction, say 5' to 3'.
Give me an example.
Okay, take GATC on one strand, read 5' to 3' on it.
Sure.
The complementary strand, 3 to 5',
Uh-huh.
would be CT-TAG, but if you're that strand also 5 to 3', it's GATTC. Same sequence.
Ah, I see. It's a kind of symmetry across the two strands.
Exactly. And restriction enzymes recognize this symmetry. They often cut within these palindromic sites, but crucially, they cut the two strands slightly offset from each other.
Not straight across.
Often not. They might cut between the G and A on both strands. In that GTC example, this leaves short, single-stranded overhangs at the ends.
The sticky ends.
This is the sticky ends. They're called sticky because, as we said, they have exposed bases that can form hydrogen bonds with a complementary sticky end.
So if you cut two different DNA molecules with the same enzyme,
They'll have matching sticky ends that can pair up. And that stickiness makes it much easier for DNA ligase, the molecular glue, to come in and permanently join the backbones.
okay, so cut source DNA, cut vector DNA with the same enzyme, mix them, add ligase, boom, recombinant DNA. Elegant.
Very elegant. It's the foundation of genetic engineering.
All right, so we've cut the DNA. Now we have potentially many fragments. How do we sort them out and get the one we want?
That's where the next tool comes in. Gel electrophoresis. It's for separation and isolation of DNA fragments.
The one with the glowing bands, how does that work? What's the principle?
Well, DNA has a negative charge, right?
Oh,
Because of all the phosphate groups in its backbone.
Okay, negatively charged.
So if you put DNA in an electric field, it will move towards the positive pole, the anode.
Makes sense. Opposites attract.
Now, you do this not in an open liquid, but within a gel matrix. Usually agarose gel, which comes from seaweed, forms a kind of mesh.
A sieve?
Exactly, molecular sieve. The DNA fragments have to wiggle their way through this mesh towards the positive electrode.
And smaller ones get through easier.
Precisely. Smaller fragments navigate the pores more easily, so they move faster and farther through the gel in a given amount of time compared to larger fragments. Size-based separation.
Neat. But how do you see the DNA? It's invisible, right?
It is. So after you run the gel, you stain it, usually with a chemical called ethidium bromide.
Okay.
Ethidium bromide slips in between the DNA base pairs, intercalates. Then, when you expose the gel to UV light,
It glows.
the ethidium bromide bound to the DNA fluoresces bright orange. So you see distinct bands, each band containing millions of DNA fragments of the same size.
Okay, so we can see bands corresponding to different sizes. How do we get our specific fragment out?
You figure out which band should contain your gene of interest based on its expected size. Then you physically cut that band out of the gel with a scalpel.
Just slice it out.
Yep. Then you extract the DNA from that little piece of gel. That process is called elution. Now you have your pure, ready to be joined with a cloning vector.
We mentioned plasmids and bacteriophages earlier. These are the workhorses.
They are, because they can replicate independently inside bacterial cells. Bacteriophages are viruses that infect bacteria. Plasmids are those extra DNA circles.
And they replicate differently. Some make more copies than others.
Yes, that's key. Bacteriophages usually replicate to very high numbers inside the host cell. Plasmids vary. Some might only have a few copies per cell. Others can have 1,500, even more. High copy number plasmids are often preferred.
Why? More bang for your buck?
Essentially, yes. If you link your foreign gene to a high copy number vector, you'll get many more copies of your gene amplified inside each bacterial cell.
Makes sense. So they act as delivery trucks and copy machines. Are the vectors used in labs just the natural ones?
Often not. Many vectors used today are engineered. They're specifically designed to make the process of linking in foreign DNA easier, and also to make it easier to find the cells that actually took up the recombinant DNA.
Okay, purpose-built tools. What features does a good cloning vector need? Our sources list a few key things.
Absolutely. Point number 1, it needs an origin of replication. ORI.
The starting point for copying, we covered that.
Right. Any DNA linked to the ORI will get replicated. And this specific ORI sequence can also control the copy number. Some ORI sequences lead to more copies being made than others.
Okay, so ORI is crucial for replication and copy number. What's next?
Second, it needs a selectable marker. This is vital.
To select the cells we want.
Exactly. It helps you identify and eliminate the cells that didn't take up the vector, non-transformants, and allows only the ones that did the transformants to grow.
How does it do that?
Usually it's a gene conferring resistance to an antibiotic like ampicillin, tetracycline, kanamycin, things that would normally kill E. coli.
So normal E. coli die if you add ampicillin.
Correct. But if an E. coli cell takes up a vector carrying the ampicillin resistance gene, it can now survive and grow on a plate containing ampicillin. All the others die off.
Clever. A way to filter out the failures.
Okay, ORI, selectable marker. What else?
Third, you need good cloning sites. These are the specific locations where you'll insert your foreign DNA.
Using restriction enzymes.
Right. Ideally, a vector should have only one, or maybe just a few, recognition sites for the common restriction enzymes you plan to use.
Right, only one.
If a vector has multiple sites for the enzyme you're using, cutting it will chop it into several pieces, making it really hard, maybe impossible, to ligate your gene in correctly. You want to cut the circular vector in just one place, making it linear so you can insert your gene.
Okay, a single convenient spot to insert the gene. Where are these sites usually located?
Often they're deliberately placed within one of the selectable marker genes.
Inside the antibiotic resistance gene. Why?
It provides another way to check if your insertion worked. Take the example of the vector PBR322. It has resistance genes for both ampicillin and tetracycline.
Okay.
Now, let's say there's a cloning site for the enzyme BAM-HI, right in the middle of the tetracycline resistance gene.
If you cut with BAM-I and successfully insert your foreign DNA there, what happens to the tetracycline resistance gene?
You've broken it. It won't work anymore.
Exactly. You've caused insertional inactivation. The cell will still be resistant to ampicillin because that gene is intact, but it will now be sensitive to tetracycline.
Ah, so you can select first on ampicillin to get all cells with any plasmid.
Right.
Then you test those colonies on tetracycline. The ones that don't grow on tetracycline are the ones with your insert.
Precisely. The non-recombinants, vector without insert, will grow on both ampicillin and tetracycline. The recombinants, vector with insert, grow on ampicillin but die on tetracycline.
That's clever, but sounds a bit fiddly, testing on two different plates.
It can be. That's why alternative methods were developed. One popular one uses insertional inactivation of a gene called LAC-C, which codes for an enzyme called boggalactosidase.
How does that work?
If the LAC-C gene is working, the bacteria make the enzyme, and if you give them a specific chemical substrate, X-gall, the colonies turn blue.
Okay, blue means no insert.
Right. But if you insert your foreign DNA into the LAC-C gene cloning site, you inactivate it. No functional boggalactosidase is made.
So the colonies stay.
White or colorless.
Oh.
So on the right plate, blue colonies mean the vector relocated without your insert, while white colonies mean you likely have a successful recombinant. Much easier to screen visually.
Blue-white screening. Much simpler. Okay, these vectors sound great for bacteria. What about getting genes into plants and animals? Do we need different vectors?
We do, yes.
That.
And interestingly, the knowledge often came from studying how some microbes naturally transfer DNA into eukaryotes.
Like hijacking nature's own delivery systems.
Kind of. Take Agrobacterium tumefaciens. It's a bacterium that infects many plants.
Causes tumors, right? Crown gall disease.
Exactly. It does that by transferring a piece of its own DNA, called tDNA, from its T plasmid, tumor-inducing plasmid, into the plant cell's genome. This tDNA tells the plant cell to make tumors and also chemicals the bacterium needs. Scientists figured out how to disarm this T plasmid, remove the tumor-causing genes, and replace them with genes we want to introduce into the plant.
So use the tDNA delivery mechanism, but swap the cargo.
Precisely. Modified T plasmids are now standard vectors for creating genetically modified plants.
Okay. And for animals?
For animals, a similar story with retroviruses. These viruses naturally integrate their genetic material into the host animal cell's DNA. Some can even cause cancer.
Like HIV?
HIV is a retrovirus, yes. But scientists have learned to disarm other retroviruses, remove the harmful genes, and use them as vectors to deliver therapeutic genes into animal cells. Gene therapy often relies on this.
So disarmed viruses as delivery vehicles. Fascinating. Okay, so once you've ligated your gene into the right vector, you need to get it into the competent host. Why do cells need to be made competent?
Because DNA, being hydrophilic, water-loving, and negatively charged, doesn't just drift easily through the cell membrane, which is fatty, hydrophobic.
The membrane is a barrier.
Significant one. Most bacterial cells aren't naturally inclined to just suck up DNA from their surroundings. So we have to treat them to make them competent, basically, temporarily, more permeable to DNA.
How do you do that? What's the trick?
A common method is treating the bacteria with a specific concentration of divalentcations. Calcium ions are often used, like calcium chloride.
What does the calcium do?
It's thought to help in a couple of ways. Maybe it neutralizes some of the negative charge on the DNA in the cell surface, reducing repulsion. It might also help create transient pores or somehow disrupt the cell wall and membrane structure slightly, making it easier for DNA to get through.
Okay, calcium treatment. And then there's the heat shock.
Yes. After you mix the calcium-treated cells with your recombinant DNA and keep them cold on ice for a bit,
Right.
you give them a very brief, sudden blast of heat, typically 42 degrees Celsius, for maybe 30, 90 seconds, then immediately back on ice.
Why the heat shock?
The exact mechanism isn't perfectly understood, but this rapid temperature change seems to somehow create a thermal imbalance or stress that helps drive the DNA uptake into the cell through those temporarily formed pores. It significantly increases the efficiency of transformation.
Ice. Heat, ice. Got it. Are there other ways to get DNA in, especially for cells harder to transform, like animal or plant cells?
Definitely. For animal cells, you can use microinjection, literally using a superfine needle to inject the DNA directly into the cell's nucleus. Very precise, but laborious.
Wow. Direct delivery.
For plant cells, which have tough cell walls, biolistics, or the gene gun method is used.
Gene gun. Sounds dramatic.
It kind of is. You coat tiny metal particles, gold or tungsten, usually with your DNA. Then you use a device to fire these microprojectiles at high velocity into the plant cells or tissues.
So you literally shoot the DNA in.
Essentially, yes. The particles penetrate the cell wall and membrane, carrying the DNA inside. And of course, we mentioned the disarmed pathogen vectors, like the modified agrobacterium for plants and retroviruses for animals. They infect the cell and deliver the DNA as part of their natural cycle.
Okay, a whole toolkit for getting the DNA inside. Now let's put it all together. The processes of recombinant DNA technology. Walk us through the sequence.
Right, it's a series of steps done in a specific order. Section 11.3 in our source lays it out.
Step by step. What's first?
11.3.1. Isolation of the genetic material, DNA. You need to get the DNA out of the source organism, and you need it pure.
Why pure?
Because restriction enzymes need clean DNA to work properly. You need to get rid of other big molecules like RNA, proteins, polysaccharides, lipids.
Cellular junk.
Basically. So first, you break open the cells, cell lysis. The enzyme you use depends on the cell type. Lysozyme for bacteria, cellulase for plants, chitinase for fungi.
Matching the enzyme to the cell wall.
Exactly. Once the cell contents are released, you treat the mixture to remove the unwanted stuff. Ribonuclease, RNase, gets rid of RNA. Protease gets rid of proteins. Other treatments remove lipids and polysaccharides.
Cleaning up the DNA.
Yep. Then the final step is often adding chilled ethanol. Pure DNA isn't soluble in ethanol, so it precipitates out of the solution.
Comes out as visible strands.
As fine white threads, usually. You can actually wind these threads onto a glass rod, a process called spooling. Now you have purified DNA.
Cool. Okay. DNA isolated. Step 2.
11.3.2. Cutting of DNA at specific locations. This is where the restriction enzymes come back in.
The molecular scissors again.
Right. You take your purified DNA and incubate it with the specific restriction enzyme you've chosen under its optimal conditions, temperature, pH, etc.
And you do this for both the source DNA and the vector DNA?
Critically important, yes. You cut both with the same enzyme to get those compatible sticky ends.
How do you know the cutting worked?
You often run a small sample on an agarose gel electrophoresis again. It lets you check if you got fragments of the expected sizes.
Verify the cuts.
Okay?
Then you join them.
Yes. You mix your cut gene fragment, the inserts,
Mhm.
with your cut vector, and then you add DNA ligase.
The molecular glue.
Exactly. It seals the gaps in the sugar phosphate backbone, creating a stable, covalently linked, recombinant DNA molecule.
Okay. DNA isolated, cut, and joined. What's next? Sometimes you need more of your gene before putting it in the vector, right?
Good point. That brings us to 11.3.3, amplification of gene of interest using PCR, polymerase chain reaction.
The DNA photocopier.
That's the one. If you only have a tiny amount of your gene of interest, or if you want to generate lots of it quickly for cloning, PCR is the way to go. It lets you make billions of copies in vitro.
Remind us of the key ingredients.
You need your template DNA, the one containing the gene. 2 primer short synthetic DNA pieces designed to flank your target gene sequence. A supply of nucleotide building blocks, DNTPs. And crucially, a heat-stable DNA polymerase like Taq polymerase.
Taq polymerase from the hot springs bacteria. Why heat stable again?
Because PCR involves cycles of heating and cooling. You need high heat, around 95 degrees C, to separate the DNA strands, a denaturation. Then cool it, 50-65 degrees C, so the primers can bind, annealing. Then heat it slightly, usually 72 degrees C, for the polymerase to copy the strands, extension.
Denature, anneal, extend, repeat.
Exactly. And a normal polymerase would be destroyed by the 95 degrees C step. Taq polymerase survives, so you don't need to add fresh enzyme every cycle. Each cycle doubles the amount of your target DNA. Repeat 25-35 times, and you get exponential amplification.
Billions of copies from potentially just one starting molecule. Amazing. Then you can ligate this amplified fragment into your vector.
Precisely. Okay, so now you have your recombinant DNA either from ligation directly or after PCR amplification and ligation.
Next step.
11.3.4, insertion of recombinant DNA into the host cell organism, getting it inside.
Right. Using one of the methods we discussed, heat shock for content bacterial cells, ectooperation, gene gun, microinjection, viral vectors, whatever is appropriate for the host. And then you need to find the cells that actually took it up.
Yes, using the selectable marker. Remember the ampicillin resistance example?
Yep. Plate the transformed E. coli on agar plates containing ampicillin.
Only the E. coli that successfully took up the plasmid carrying the ampicillin resistance gene will survive and grow into colonies. The untransformed ones die.
So the antibiotic resistance gene acts as the selectable marker, letting you select for transformants.
Exactly.
Okay, we've got our transformed cells growing.
Yep.
What's the ultimate point? Usually making something, right?
Usually, yes. That leads to 11.3.5, obtaining the foreign gene product. The whole point is often to get the host cell to produce the protein encoded by your inserted gene.
The recombinant protein.
Correct. For this to happen, the recombinant DNA needs to be expressed. The gene needs to be transcribed into messenger RNA, and that mRNA needs to be translated into protein by the host cell's machinery.
Does that just happen automatically?
Not necessarily. You often need to make sure the gene is inserted into an expression vector, which contains the necessary control sequences, like promoters, that the host cell recognizes to turn the gene on.
Okay, so you need the right signals for the host cell to read the gene.
Yes. Assuming it's expressed, you first grow the cells, maybe on a small scale initially, to confirm production. Then you break open the cells, if the protein stays inside, and use various techniques to purify your desired protein away from all the other host cell proteins.
Sounds complex.
It can be. And if you need large amounts, you move to bioreactors.
Those big tanks.
Right. Vessels from maybe 100 to over 1,000 liters, where you can grow huge cultures under tightly controlled conditions.
What conditions need controlling?
Everything critical for growth. Temperature, pH, oxygen supply, if aerobic, nutrient levels, substrate, salts, vitamins. The goal is to maximize cell growth and production of your protein.
You mentioned stirred tank reactors are common.
Very common. Usually cylindrical with a curved base for better mixing. They have an agitator system, basically a stirrer, to keep everything mixed and ensure oxygen gets distributed. They might bubble air or oxygen through too.
And lots of sensors and controls.
Oh, yes. The systems for foam control, temperature control,
Yeah.
pH control, sampling ports to check on the culture without contaminating it. Some run as a continuous culture. You constantly drain off used medium and add fresh medium.
Why do that?
It keeps the cells in their most active growth phase, the log phase, which often leads to higher productivity, more protein yield over time compared to just letting a batch grow and stop.
Okay, large-scale controlled production in bioreactors. We've made our protein. Are we done?
Not quite. The final stage is 11.3.6, Downstream processing.
Downstream, meaning after the bioreactor.
Exactly. Everything that happens after the biosynthesis phase before the product can be marketed or used.
What does that involve?
It starts with separation and purification, getting your protein away from the cells, the culture medium, other proteins, etc., and purifying it to a very high degree.
How pure?
Depends on the product. For therapeutics like insulin or vaccines, it has to be incredibly pure. Then comes formulation.
Formulation, like making it into a pill or injection.
Sort of. It's putting the purified protein into a stable, usable form, often adding preservatives or stabilizers so it doesn't degrade. For drugs, this stage also involves clinical trials to test safety and efficacy in humans.
Right. Essential for medicines.
And throughout downstream processing, there's strict quality control testing. Every batch has to meet predefined standards. It's a complex, often expensive, but absolutely critical part of the whole process.
Separation, purification, formulation, trials, quality control. Okay, that brings us to the final product. It's quite a journey from finding a gene to having a product.
It really is a testament to understanding and manipulating biological processes at a very fine level.
What strikes me is the precision needed at every single step, from choosing the right enzyme to controlling the bioreactor pH.
Absolutely. It combines deep biological knowledge with sophisticated engineering principles.
So to quickly recap this deep dive, we started with the historical context, this anthropocentric view driving technology.
Right, Descartes and the focus on human welfare.
Then defined biotechnology, broad traditional uses like fermentation, and the modern, more restricted sense involving genetic modification.
And the EFP definition integrating natural science and organisms.
We identified the two core pillars: genetic engineering, altering the genetic material itself.
And bioprocess engineering, maintaining sterile, large-scale conditions for production.
We traced the conceptual path to genetic engineering, contrasting sexual-actual reproduction, the limits of traditional hybridization.
And how recombinant DNA overcomes those limits by isolating only desired genes, needing an ORI for replication and cloning.
Then the landmark Cohen and Boyer experiment, restriction enzymes, plasmids, ligas, creating the first recombinant DNA and cloning an antibiotic resistance gene.
Which established the basic steps. Identify DNA, introduce to host, maintain, and transfer to progeny.
Then we unpack the toolbox: restriction enzymes, molecular scissors, their discovery, naming, palindromic recognition sites, creating sticky ends.
Facilitating ligation. Then gel electrophoresis for separating fragments by size, visualizing with ethidium bromide, and eluting the desired band.
Followed by cloning vectors, plasmids, bacteriophages, their copy numbers, and the essential engineered features. Origin of replication, selectable markers like antibiotic resistance or blue-white screening via insertional inactivation.
Cloning sites and specialized vectors for plants, Agrobacterium-type plasmid, and animals, disarmed retroviruses.
We looked at making hosts competent calcium treatment, heat shock, and other delivery methods like microinjection and the gene gun.
Right.
And finally, we walked through the entire process step by step, isolating pure DNA, lysis, enzyme treatment, precipitation.
Cutting DNA with restriction enzymes, potentially amplifying the gene with PCR, denature, anneal, extend with Taq polymerase.
Inserting the recombinant DNA into the host and selecting transformants.
Obtaining the foreign gene product via expression in bioreactors with optimized conditions.
And finally, the crucial downstream processing, separation, purification, formulation, trials, and quality control.
That covers the core principles and processes pretty thoroughly, based on our source material. Understanding these steps really unlocks how biotechnology has reshaped medicine, agriculture, and industry.
It's undeniably powerful, and having dissected it like this, you really appreciate the intricate science involved. It certainly makes you think, doesn't it? We've talked about the how, but maybe it leaves you, our listener, pondering the what next, or even the should we, regarding specific applications, like how far will gene editing take us, or what are the ethical boundaries of using these incredibly powerful tools? Food for thought, perhaps.
Definitely. The potential is immense, but so is the responsibility.
Indeed. Well, thank you for joining us on this very detailed deep dive into the principles and processes of biotechnology.